Okay, here we are talking about our next cell process, which is going to be important for this year and also moving forward and understanding how cells function and survive and whatnot. So, what we're going to talk about today is diffusion and osmosis. And then we're going to talk about how that applies to how things move across the cell membrane. What we're going to be mostly focusing on here is simply how certain particles will be able to move by diffusion osmosis, and we won't get to a huge amount of differences about other methods of going across the membrane. Now, the first thing we're going to go into here is that cells live in a watery environment, so it is a solution where water is the major component. It is the thing that is dissolving all the things that move around, whether it's your blood, it's the space outside of cells, the extracellular space, the interstitial space, or inside of cells. So we're going to go through some basic components of a solution, and they are listed here for you, but we'll highlight what they are. Now, solutes are things that are dissolved in some, usually a liquid, doesn't have to be, but generally it's going to be a liquid. So solutes are the things that get dissolved. And since we're talking about living systems, our solvent, the thing that is doing the dissolving of these different types of solutes, is water. So the thing that is doing the dissolving, so things that are dissolved inside of a liquid, or the liquid that is dissolving those things that are now inside of it, is the solvent. So things that are dissolved in something, so the thing that they go into is the solvent. In biological systems, the solvent is water, and anything that's inside of that water is essentially going to be a solvent. Okay? Anything that goes in or out of a cell are called solutes generally, and the water is the solvent. Now, diffusion is kind of how these solutes, now that they're in solution, how do they move around, I suppose, because cells are not static. There's a lot of dynamicness to them, and these different particles that are in there are going to be important for them to survive. So these different solutes could be like glucose, which is an energy source. It could be proteins, amino acids that are going to be used in help creating things for the cells. So it's important for things to be able to move, and that's one role that the solvent plays. So within the solvent, solutes are able to move around. And the way that they move around in a solvent is by randomly colliding with other solute particles. And when particles that are dissolved in solvents, these solutes move around because of collisions, that is called diffusion. All of the particles that are dissolved in that solution are constantly and randomly moving. As a result, over time, the particles will tend to spread themselves out evenly. That's because they're always moving. Now, diffusion is a description of the tendency of these particles to move from an area from a high concentration to a low concentration of the same particle. Okay? So that's important. So diffusion, when you observe things that are dissolved and able to move around in some sort of solution, they tend to move from an area of low concentration to an area of high concentration to an area of low concentration. Okay. To an area where they are less concentrated. Okay. Now, this is because of random collisions. So what that means is, if you looked at the diagram below, so all of these solute particles that are the water is not labeled on here, the solvent. You would assume all the white here, everything that's not kind of a particle is going to be represented by water, even though water is technically a particle, but we're going to leave the solvent out and just talk about the solute. So these are the solutes here. They're going to be randomly moving, and they will bump into each other. Now, if you're in the middle of this kind of clump here, you're going to be bumping against your own particles a lot. But the ones on the outside, they're going to be, once they bump and collide, they can be pushed away and start to spread out. And as they start to bump and collide and then move away from those particles, then it opens up the freedom of these other ones to move around and move away from another, and so on. So they will tend to bump into each other a lot when they're all together, but over time, since they're colliding and some are moving away from them, they will tend to spread out. So they will tend to go from the area where they are high concentrated to an area where they're low concentrated. Now in this diagram, there's actually none over here, so that's a very low concentration of this particular solute. So as these ones collide, they're going to be bumping around, and some are going to move over here. Okay. All right. Now they will continue to do this until there is an equal concentration of that type of solute or particle in the areas of which they can spread to. So in our diagram, we have two areas here, one and then two. Over time, they will continue to undergo diffusion until they are equally spread out. Now they will still be at motion and moving around, but because they are not increasing on any side, so they're not spreading out over here to go over here, and they're not spreading out over here to go over here, we say that the movement now is at equilibrium. So they're evenly spaced, that the number of collisions on both sides will randomly push the same number to one area and then the same number going back to another area. You won't see a concentration change. Okay. All right, so let's go back to the diagram over here. These ones over here currently separated by the cell membrane from over here. So this is inside the cell, this is outside of the cell. These are high concentration. They will collide much more frequently, and some of them will end up getting bumped, and some of them will move across the membrane. Okay, so you can see a few of them have moved over here. These will still be a high concentration area compared to the inside, but you can see that the concentration over here is increasing, and the concentration over here is decreasing. And this will continue to happen until they are at equal concentrations. They will still bump and move across. They don't stop bumping and moving across, but the rates will be equal. These ones going over here and these ones going over here will be the same, and you won't notice any differences in the amounts of each of these on those different sides. Okay? Now, when things move from here to high to low, and where you're actually increasing those numbers, that's kind of a net movement. So in this diagram, there's a net movement of particles into the cell because they're physically increasing in number over here compared to what they were before. When you reach this situation, equal concentrations, we say that the particles are in equilibrium. Which means that there is no net movement into or out of the cell here. It's equal. So the rate of going in is equal to the rate of going out, and you won't notice any amount difference in the solutes between the two sides. Okay? So diffusion is going from a high concentration area moving to a low concentration area, and if able to, will stop when the concentrations are equal, net movement wise. But the diffusion will, I guess, still technically takes place but there's no net movement, it's at equilibrium. Now, for diffusion to take place, we need that high concentration and a low concentration, and that is called a concentration gradient. So a gradient is a difference between two things, three things, or just something between at least two areas. To have a gradient, one needs to have a high amount of something, and the other area needs to be a low amount of something, or nothing, and so on, which would be a low amount. And moving along a concentration gradient, something that occurs when we talk about diffusion and osmosis. So moving along a concentration gradient means that things are moving from a high area of high concentration to an area of low concentration. This is referred to as being a type of passive transport. Now that doesn't really affect you in this course, but it will come up in grade 12 or maybe AP 11. And passive means that no energy is expended by the cell. In order to accomplish that movement. So the diagrams down below, they're kind of showing you a type of gradient. Now, when you probably hear about gradients in your life, you're probably talking about things that are graded, which is usually like things that you have to walk uphill, downhill. When you introduce grades into things, it means they're a little bit higher and so on. Okay? So if you looked at these two diagrams, you can see that this has a higher gradient than this one, right? Um, it's going to be moving faster. It's going to get from top to bottom much quicker, top to bottom much quicker. But the whole idea here is not necessarily their size. It's just that if I was to put something up here, let's say a ball, it would passively roll down this hill. Okay? And because of gravity, and it's round, so there's low friction on it here. There's not enough surface area for it to connect, so it's going to start rolling down by itself. Okay, so that would be passive. If you look at this one here, it's a little bit steeper in that it doesn't have as much distance for the rolling down to go, but the same thing would occur. This is also going to be passive starting at the top here, right? And this represents the high concentration here. This represents a high concentration over here. And then this ball will roll down or diffuse down its gradient. It will move along the gradient down the ramps. Now, if the ball starts down here, you can actually get it to go to that up there, but that would not be passive anymore in order to go that way. And that's something you kind of cover in grade 12. And up here, it's not passive. We said that going down here would be passive. It doesn't take any energy when you go from high to low. But this would be kind of moving from low, going to high. This one would be starting off as low concentration and going to high. In order to go or move my particle from a low concentration to a high concentration, it's not going to be passive. I'm going to have to spend energy to do that. Okay? And that's something you cover in grade 11 and 12. But for now, we will just talk about diffusion, moving along its gradient, going from areas of high concentration to areas of low concentration, with our analogy here, going down a ramp. Now, in order for diffusion to take place where you have net movement, you have to have that gradient. Okay? These technically we don't really say they're diffusing, although the particles are moving. So this would be kind of the diffusion over here. This would now be equilibrium. They're not diffusing anymore. And they're not diffusing because there is no gradient. Gradient here, there's a gradient here, but because these are equal, there's no gradient anymore. So diffusion stops at that point. Okay? In order for diffusion to take place, you must have a concentrate or some type of gradient. Okay? It is the driving force between many molecules or atoms or ions moving in and out of cells. It's not the only thing that particles move in and out of cells, but there is a number of them that do move by diffusion. Now, this is something that comes up a little bit in grade 12, so we're just going to quickly go by this as we jump over to osmosis. Actually, we'll kind of skip this part here. You can read it through. It'll be something that comes up in grade 12. Let's just go over to osmosis here. Now, osmosis is just like diffusion. So it's technically it is diffusion, because it will still follow the idea that there must be a gradient in order to have osmosis occur, just like diffusion did. However, osmosis is the movement of water across a cell membrane, or what we will say a selectively permeable membrane, along water's concentration gradient. And the interesting thing about water, in the previous example of talking about those solutes, water is a solvent. So when you look at biological systems, for the most part, the cell membrane, which is this selectively permeable thing, it blocks many things. It's a barrier into and out of the cell. So there's not a lot of things that have free movement through the membrane. So particles usually don't diffuse through a cell membrane. They can diffuse inside of the cell in different locations and stuff like that, but usually not through. Some can diffuse through, but not a heck of a lot actually diffuse through the membrane, because your cell wants to maintain an internal environment that's conducive to survival. So it's going to block and create a barrier for many things to get into or even out of the cell if they want to keep those things inside of it. So, but what can move readily by diffusion or by a concentration gradient is water. And so osmosis is going to be how a solvent is moving in and out of the cell by diffusion, and usually you don't see that from solutes because they are controlled the way that they enter. So we're going to look at how water moves inside and outside of cells. Okay? Now the cell membrane is permeable, which means it can enter, so can move through. You'll learn the details about why that is in future courses, but for now we'll just say that you can move through it reasonably easily, but not a lot of types of solutes do, which I mentioned before. So you get a lot of water going in and out, but solutes have to be controlled by the cell membrane. Osmosis is a type of diffusion, but instead of water moving, we talked about this before, it's going to be water, instead of solutes moving, it's water moving, and the water is going to be moving along a gradient, so it's going to be going from its high concentration to low concentration. Okay, so its water moves from an area of high to an area of low. So essentially the same criteria as all other diffusion, except now it's water moving along its gradient of different amounts of water. Now the diagram below, I like to mention this to my kids. You notice that it's shaped like a U, and it's a tube. So this was the original U tube that chemists have had for, I don't know, hundreds of years. So you think you're pretty cool with your U tube, but there was already a U tube in use by chemists. And this is one of the things that they used the U tube for, was to do some research into pressure, and they also use it for selective permeable membranes and so on. But anyway, here we are with this tube that has water and solute in it, and then there's this selectively permeable membrane that separates the two sides of the U tube. And you'll notice that these dots are representing the amount of solute. This other stuff around is representing water. So if you looked at this diagram over on this side, it's the same amount of water as this other side, but there's more of these dots in that one. So if there's more solute in the same amount of volume, this side is what we can say has a high solute concentration. So the brackets represent concentration. Something you learn in chemistry. The arrow is just showing that it's high, it's going up, so it's high solute concentration. If you compare it to this side, so same volume, so if we're looking at pure concentration here, this is a lower solute concentration to that side. And then nothing, these particles are too big to move through this membrane. So this is a barrier or blocking that we talked about before that the membrane does. So these solutes are restricted from moving. However, water can move, it can diffuse through this membrane, and simply because of the water colliding with itself, because it itself is particles, what will happen over time is you'll notice that the two concentrations will become equal, but in order to achieve that, the water has osmosed to the side where there was less water. Okay? Now, in order for us to predict what that's going to look like, we have to make, so this is a high solute concentration, this is high solute. Now, since this is an amount of water, it's a ratio of water to volume. So in this one, I have a lot of solute, which means there's going to be a high concentration of it compared in the same volume. But at the same time, if I have high solute, I'm going to have low water concentration. So if my solute concentration is high, or amount is high, I'm going to have low water amount by comparison if the volumes are the same. Now, if I look at this side, if I want to make a statement about the water, it's that if this was high solute, it ended up with having low water concentration. If this is low solute concentration, I'm going to have a higher water concentration because our volumes or amounts are the same. So I have more water by comparison to the solute. I have less water in comparison to these solutes here. And as a result, what we want to identify is the area of water concentrations. Okay? And it will diffuse in osmosis. So visually from this, we can get the solute concentrations, and from that, we can get the water concentrations. And now we've established that this is high water. Sorry, this is high water, and this is low water. We know that the water will diffuse from this side to this side. Okay? We'll move from here to here through that selectively permeable membrane. So the water will be moving through here. And you would notice that if you had one of these U tubes with a selectively permeable membrane and different solute type or a solute type in here, you'll notice that the water goes up because it's moving into that side. This water goes down because it's going from here over to here. Kind of interesting. Now, how does that apply to cells? So the reason that it's relevant to cells is because cells live in an aqueous environment. So their environment around them is water, the environment inside of them is water. So water is always there. It is the solvent dissolving everything. Their cytoplasm is a solution composed of water, things dissolved in it, as well as its surroundings. Because of osmosis, cells are sensitive. So keep that in mind. Cells are sensitive to the water content of their surroundings. So that means outside of a cell. And they can be significantly affected by severe changes in those concentrations. Now, we'll talk about some severe situations in our work coming up here. Okay? All right, so there were 3 general types, now depending on the severity of them, well, for 2 of them anyway. So there's a hypertonic solution a cell could be in, a hypotonic solution a cell could be in, and then an isotonic. All right. So these diagrams represent what happened to red blood cells. So all of these cells in here are red blood cells. Now the file that you have is better because it's in color. Mine is just a printed off version so I can make these additional notes. But you'll notice that these have kind of a unique shape. If you looked at them from the top, they have this little divot in the middle there. And if you looked at them from the side, they have this unique kind of shape like this. And we'll get to that later when we get to the circulatory system. But this is called biconcave. And it allows red blood cells to function the way that they do. And it's important that they have this shape because they will lose some of that functionality if that shape is not maintained. Okay? So we'll keep that in mind as we look at these different types of solutions. Okay? Now, ideally, anything that keeps the shape the same is good. So if a shape does not change, that's a good thing. Now, just like our activity with surface area and volume is that shape is an important thing for cells. And in this type of cell, shape is very important. And you don't want it to change. So not changing a shape is a good thing. Shape changing would therefore be a bad thing. Okay. All right, so let's look at this hypertonic solution over here first. Okay. A hypertonic solution is a solution that has a higher amount of solute. They have high solute concentrations compared to the cell. Okay. So in the diagrams above, we'll add some additional ones here in just a moment. Okay. This means, so hyper means kind of like more. So I don't know, kids used to be, that had a lot of energy, used to be called hyperactive, and the hyper meant they have a lot of energy. So hyperactive, lots of energy. Hypertonic means lots of solute in that solution. Okay? And we're going to add some drawings up here to represent that. So let's say I have my original cell. Here's my cell. Okay. Now what I'm going to do is going to put high solute outside of the cell. Okay. And since this is a comparison to the inside of the cell, if this is high solute outside, that means that inside the cell, it's low solute. Okay. Now, like the YouTube activity that we did, if this is high solute, since it's a ratio between solute and solvent, this means that by amount, this is going to be a low water. And since this is the same thing here, if this is low solute, this is going to be high water. And then once we have that, we want to establish where there is high and low water, and then we will see how the water diffuses through that selectively permeable membrane. And you'll notice that there's a high water outside of the cell, there's low water on the inside. So water will diffuse, the other way. There's high water inside, low outside, so water will diffuse out of the cell. Now, when water is leaving the cell, that means that the cell is going to get smaller, and you end up with something like this. Okay? The cell shrinks. Okay? Because water is leaving the cell. Okay? This changes the shape. Okay? And we talked about this before. Shape is important for these specific types of cells, as it is for all cells. So how shape change like this is bad. You don't want your cells to shrink. Okay. All right. So water replaced moves out of the cell. Water moves out of the cell and leaves uh, because the water concentration gradient. So it shrinks in size, and now we call this crenated. So this cell has become crenated, okay, or it's undergone crenation, or it's being crenate. Okay? But usually we just say it's crenated. Okay? That's a special fancy word in animal cells for shrinking in size. All right, let's look at the hypertonic situation over here. So hypertonic is the reverse of hypertonic, or hypotonic is the reverse of hypertonic. So hyper is more, hypo means less. So the outside of the cell has a lower concentration of solute, and we'll add some drawings to reflect that over here. So if we did our drawing again for our red blood cell, there we go. So now outside is low solute, and by comparison, this would be high solute. And then since these are proportional for concentration, if this is low solute, this means it's high water. If this is high solute, this means that it's low water. Then we establish where there's high water and low water to see if there's a gradient, and there is. And then we have the water travel from high to low, so high to low. So in this case, the water will be traveling through the selective permeable membrane into the cell. And instead of shrinking the cell, it's going to cause the cell to swell, okay, which means it gets bigger. All right. Placed in the hypertonic, water moves into the cell, caused by that gradient we talked about from the surroundings. There's a net movement of water inside of it, making the cell bigger, swollen. And then animals are susceptible by getting too large because they can undergo something which is called lysis. And what lysis means is the cell bursts. And if a cell bursts, the cell dies, and then you've lost your cells. So that would be a bad thing. So it would be like a cell like this, then it swells, and it's possible it swells so much it bursts, and everything flies out, and your cell is effectively dead. Okay? That's lysing. Now, an isotonic solution is one that has equal solute concentration with the inside of a cell. So there's no gradient, which means no osmosis occurs. There's still water moving in and out, but it's equal rates. So we will say that there is no osmosing here, although technically it is going in and out at the same rates. There is no net movement of water. Therefore, the cell shape does not change, the size does not change, and so on. So this would be the ideal solution for your cells to be in, because both of these two types of solutions will cause the cell to change its size and shape, and that's always bad for cells. The plant cells go through a slightly different process because they have a secondary, or they have another organelle that's called the cell wall. So they respond differently to these solutions. They still have kind of the same effect, but they have something that will prevent them from lysing. Okay? But they have these cell walls. So cell walls prevent cells from lysing in a hypotonic situation. Okay? So they prevent lysing. Cell walls are rigid, and they're not really barriers. They're just a hard or a rigid structure on the outside that gives some structural support to plants in their bodies and so forth. Okay? But it's not a barrier. Things can move in and out of it between a cell wall and the cell pretty easily. Okay? Now, the names for the thing osmosis that happens to cells is different. So, a hypertonic environment for a plant, the same result is the same. The result is the same as the animal cell. Water leaves the cell, but because the cell wall is still there, the physical space that the cell occupied is still the same, but the cell on the inside of that wall has been reduced in size. Now, instead of saying crenated, we call that plasmolyzed, or has undergone plasmolysis. Okay. In a hypotonic environment, when water is going into it, instead of lysing, because they can't lyse because of the cell wall, they become turgid. And that is related to turgor pressures and so on. So the more water and pressing on this cell wall from the expanding cell is called turgor, and we'll just say that's turgid because it's kind of gone beyond its regular size. Um, the regular size cell here, you don't really have to say much about it. And this one here, it's called flaccid for the most part. Okay. All right, so when there's net movement into a cell, it becomes turgid. In an animal cell, it swells. And perhaps maybe lies. When there's net movement out, the plant will undergo plasmolysis or plasmolyze. In an animal cell, it will crenate.